Welcome to the Biotech Startups podcast by Exceeder. Join us as we speak with first-time founders, serial entrepreneurs, and experienced investors about the challenges and triumphs of running a biotech startup, from pre-seed to IPO, with your host, John Chi.
My guest today is Kate Yen, founder and CEO of Auron Therapeutics. Auron Therapeutics is using cutting-edge science to develop next-generation therapies targeting the dysregulated cell states of cancer. By combining a proprietary database of single-cell genomic data with an AI-powered computational platform, Auron compares healthy cell development to cancer cell development and maps out the drivers of tumorigenesis. Kate has a Ph.D. in biological chemistry from UCLA, where she also completed her postdoctoral fellowship. Before founding Auron, Kate held a position at Merck and a professorship at UCLA. She also held senior roles at Agios Pharmaceuticals, where she led the IDH Translational research team responsible for the preclinical and clinical development of 2 FDA-approved IDH mutant inhibitors. She also co-led the discovery of vorasidenib, a promising glioma therapy currently in clinical development. With Kate's extensive preclinical and clinical expertise, a proven track record of bringing innovative therapies to market, and a passion for tackling complex oncology challenges, her journey and insights are not to be missed. Over the next 2 episodes, we'll hear about Kate's journey, from her early passion for science to groundbreaking cancer research and leading Auron Therapeutics. We'll discuss how her hands-on approach shaped her career from Merck to Agios, where she helped advance novel cancer therapies through clinical trials. And we'll dive into the leadership lessons and strategic insights that now drive her mission at Auron to push the boundaries of oncology and beyond. Today, we'll chat about Kate's early days. We'll start with her time in academia, from graduate school to her transformative postdoc experience, where she developed a passion for translational biology. Then we'll dive into her transition into industry. First at Merck, where she gained invaluable drug discovery experience, and later at Agios, where she played a pivotal role in advancing innovative therapies. Along the way, we'll discuss the key lessons she learned about leadership, collaboration, and navigating the fast-paced world of life science. Without further ado, let's dive into this episode of the Biotech Startups podcast. Kate, it's so good to see you again. Thanks for coming on the podcast.
Thanks for having me. It's great to see you as well.
So, in our traditional opener for the Biotech Startups podcast, we always like to go all the way back to your early days and really learn about what kind of influenced your business philosophy, your leadership style, and frankly, what got you into science. We always love learning about the origin story. So can you take us back? Were there any, tell us about your upbringing. What was it like, in your adolescence?
Yeah, so I have 3 sisters, so we have a big family, and my dad was a scientist, and my mom was a writer, so she was more on the creative side. And we moved around a lot when I was young, so we lived not only in the U.S. but also moved to Europe for a few years and stayed there, and then came back to the U.S. So that was kind of exciting to be able to move around and see different cultures and different things. But for the majority of my childhood, I grew up in New Jersey with my parents and sisters, and was always interested in science. My grandparents were physicians on my mother's side, and so I was always intrigued by that. My dad was an engineering geologist, so I was always kind of drawn to the sciences.
Very cool. Where in Europe did you live for that period of time?
Yeah, we lived in London. My dad was actually British, so we spent some time back there. He was working all over Europe, working on engineering projects, so that brought us over there.
Amazing. Before I would hit record, I mentioned how I haven't left Berkeley, so I'm jealous that you had an opportunity to live overseas. I always wonder. I'm
yes.
What I'm probably missing a lot. Kind of.
It's a big world.
Yeah, it's a big world, and I was I got to get out there. So it sounds like you had both science and also English and literature kind of present in the household. Was there for your father, was he always you need to go into science, or was it something that was always kind of intrinsically came from within?
I think it more came from within. It was interesting. It was probably more from my mom, not in science, but was more of, the generation of, she had 4 daughters, and she wanted us to be educated and have careers and be able to do anything we wanted to. But I always liked science. I liked medicine. I thought I was going to be a doctor. I kind of thought I wanted to be an MD for all my life. And then I started to do some research in college and in, after college. And, I really got the bug and decided I thought I'd rather do a Ph.D. track.
Awesome. And you mentioned you kind of were going in thinking about, potentially being a physician. And also like your choice of university, like how did you, choose, end up going to College of St. Elizabeth? And can you talk a little about those, going in, did you have an undergraduate lab experience?
Yeah, so it was interesting. I stayed in New Jersey to go to College of St. Elizabeth mostly because my mom had come down. She was ill. She actually, sadly passed away of early onset Alzheimer's, and I was helping my dad kind of somewhat take care of her. So I stayed in New Jersey, went to school there. But I think that also kind of instilled in me the interest in research and in science and medicine. And so, it was sort of a forced choice, if you will, as to where I went to school for undergrad. But then when I went for grad school and I made the decision to do a PhD rather than an MD, I really was excited about the opportunity to kind of branch out and go somewhere new. And UCLA seemed really exciting to me.
Awesome. And did you get the lab bug while in undergraduate? Or
yeah,
was that later?
I did. I didn't. The research when I was back in college is much different than the research is now.
Yeah. Totally.
A lot different.
Yeah.
But I did. I worked in an industry. I worked at it was a company called Allied Signal at the time. I did it in the summer, and I worked and did some molecular biology research. And then during the year, I had an independent research program in my undergraduate, which really got me excited about just basic biology and sort of disease areas. But then I also went to Princeton and worked for a year in between, deciding between med school and graduate school. And while I was there, I was, the lab manager, but I also had my own independent research lab or project that I got to work on. And I was working directly with a postdoc there, and I really enjoyed it a lot. I started taking classes. I was, sitting in on some of the graduate classes, learning about the studies I was doing, and that really solidified my bug for going into doing a PhD.
That's amazing. I'm always curious about that first like jump into the lab. For me, it was like, it was just a friend, just like, hey, come check it out. I was like, what is this? I was like, sure, like, okay. And then after that, I was like, just like hooked. I was like, oh, you mean I don't have to just like memorize this textbook?
Yes.
Like, I can actually work with my hands. Like, was that similar? Was that a similar experience for you?
Yeah, no, for sure. I think, sitting and memorizing things is kind of, boring, but, you getting into the lab and reading about stuff and getting excited about ideas and thinking about how you could test those ideas and having the tools to do that in the lab to really, discover new biology, really I found fascinating and sort of charting new frontiers, which was exciting.
Very cool. Yeah. You talk about the tools, like, yeah, like even like within the past like 10 years, the tooling is like so different.
So different.
I was like thinking about like how much pipetting I did. I was like so much. And it's like, they're just robots that do it now.
I know.
I'm like, you have it so good. Like you have it so good. It was sweltering hot and just like, this is awful.
I tease all the kids so much about this now. I mean, I'm like, oh, you can just pour a gel of a different percentage. And they look at me with wide eyes like, what are you talking about?
Yeah, you're just like, what do you mean? You're like, you're looking at me crazy. Like, it's like, it's a very different time. Okay. Like this, like, understand. Whose lab were you in Princeton? Were you like were you just like basically in that postdoc lab?
No, I was working in a lab I was directed by Mike Cole, and he was the PI, and then there were postdocs within his lab, and we were studying MYC there. He has a lot of research on c-MYC, trying to understand the role it plays in basic biology and transcriptional regulation, and that's where I started my sort of hardcore molecular biology research, I would say. So it was really exciting. And actually now here I am all these years later. I ended up studying MYC in my PhD or my and then now in my company, it's coming full circle. So I feel it's going to be probably my life work here to figure out what MYC is doing.
I love how that happens. I love how like there's like these like seeds that are just like planted in like very, very early days. And I'm curious about that, like, your first, you mentioned like hardcore, like lab experience. Can you talk about like, what was a moment for you where it was like, oh, this is like another level. This is hard. And like, how did you handle that?
Yeah, no, I think, well, there were probably, technically, there was just much more sophistication to the experimental design. I mean, we were using radioactivity. I'd never used that in undergrad and things like that. And then, when I was, we're taking some of the classes, just the questions and the way they were, the tests were set up is much different than undergrad, right? It's a lot more, you have to one sentence to figure out how to solve this problem. And you're like, okay, I got to really think this through.
Yeah.
So it's really fun.
So I was just going to say, just like, I had just like, it just, and I'm sorry for interrupting you, but it's like, I had that same moment where I'll just like, it almost is like you just get thrown into the deep end.
Yeah.
And they're just like, swim. You're
yeah,
like,
exactly.
What? But sorry, I interrupted you.
No, that's exactly the same way. And, I like that, being, tackle, you get a problem in front of you and sort of figuring out how to solve it, but, tapping into expertise around me and really, finding people that have knowledge about this problem and how can we kind of crack this nut together and really working in a collaborative way. And I think that's one of the things that I've taken with me through my entire career is, no one's the smartest person in the room, and you kind of just have to figure out how to study biology. It's very complex and there's a lot of different angles. And so, working together, hopefully we can make some progress.
Absolutely. And I love hearing that, where you like that you had an opportunity to be in like a very collaborative lab, like to begin with. Because like I've like definitely done rotations where the lab is just like almost like independent work. Like
yeah, definitely.
There's different, like obviously different styles and different ways to approach it. But I find myself like I'm much more collaborative in nature.
Yes. Yeah.
Like I'm not smart enough to just do this like by myself. Like I'm not that smart. But I love that, it kind of sounds like it was like a team effort more than anything. And so, you mentioned like, this was like, you've caught the bug and you're starting to think about your graduate school. Like what schools, I know you ultimately ended up at UCLA. Were you looking elsewhere too? Or were you like, UCLA is the one?
No, I applied to a bunch of different schools. I liked, I all over the place. I liked at UCLA for a specific reason because they had just started this program called Access, where UCLA is a huge school, and we were the first class of the Access program. And basically, you came in under an umbrella through the graduate program, and you had access to any department on UCLA campus. So you didn't have to apply to just neurology or molecular biology or biochemistry. You could just go under this umbrella, and then you could do your rotations through any department you wanted. So you had so much opportunity to think through and learn and figure out what you actually wanted to do. So that was really why I decided to choose UCLA over the others.
That's amazing. I love that, because like, sometimes you can like, at least when I was at Berkeley, I was like very much in like my toxicology lane. I was just there.
Yes.
I was in one building talking to one group of people all the time. So very cool to hear that there's like the ability to just like go around.
Yeah.
And question, LA, going to the West Coast, that must have been like also a personal change for you. How was that? Like going from East to West?
Yeah, that was tough, actually. It was definitely a different experience. I've grown up mostly on the East Coast, so I much more used to this, going out there and understand this weird no seasons thing. Why is it warm when it's supposed to be Christmas? I didn't get that. But you're also in grad school, and I was fortunate because within that umbrella, you gain access to a lot of people and you meet friends and so forth. But, it was pretty isolating. All my friends and family were back East, and I was really solo by myself out there. And grad school, you're a starving grad student. You don't have a lot of money. So you're kind of trying to make ends meet. It's not exactly the cheapest place to be.
Yeah.
I got used to it once I settled in. It's definitely took some time.
Yeah, absolutely. I can imagine just being utterly confused that it's like 65 like all the time.
Yes.
And you probably like pack the wrong things. You're just like, I have all this winter gear. This is like this. I don't need this.
Yes.
But can you tell us a little bit about the lab that you're in and the research that you ended up pursuing?
Yeah. So my lab that I did my PhD in was with Mike Carey, and we were studying transcriptional regulation there. And I think I probably landed there just because, from my background working on MYC, it's a transcription factor. I kind of was in the same zone. But Mike was also is an amazing teacher, and he taught some of our early molecular biology classes. And he just made it very clear to me and organized, which I liked. And I really wanted to jump in and kind of solve that problem. So after my rotations, I decided to join his lab and really dig into how genes are turned on and off, because ultimately I wanted to apply that to translational research where, for medicines. And so, my love or my idea of thinking about treating patients and being working with patients, was always underneath there, but I was just going to apply it in a different way as opposed to being a medical doctor, do it through research that can bring medicines to patients.
Very cool. What was this lab also as collaborative as the Princeton lab?
Yeah, it was very collaborative. He's a tough advisor. He's great. I mean, he learned so much from him. But definitely he expected a lot from his grad students and postdocs and pushed hard. And but I learned a ton from him, like how to do really good experiments. I also learned how to always have something cooking in the background in case it doesn't work.
Yeah, that is a very valuable lesson. Like even outside of the lab. It's like you're talking about how these kind of lab experiences sometimes just get thrown in the deep end. But there's another element of too that's stuck out to me as you're describing this, is that when I found I grow the most when one, you get thrown in the deep end, and then there's a very high bar that's set.
Yes.
And it's terrifying. It's you're like, what? But it really stretches your imagination of what you think is possible.
Yeah.
And then you reflect through it. At least for me, I'll look back, I'm like, that was painful as hell. But there's a lot of growth that kind of came of it. It's and you always surprise yourself when you push yourself in that way. But very interesting. Yeah. And when it sounds like Mike was kind of set that bar super, super high. As you were kind of doing wrapping up your PhD, did you know and graduating from Mike's lab, did you know that you're ultimately going to go do a postdoc? Or at that point where you're thinking about what's next for me?
No, I definitely wanted to do a postdoc, but I think I always wanted to go into industry, which was not usual at that time.
That's what I was going to say.
Yeah, it's definitely not usual. I mean, it was very much kind of frowned upon. If you're in an academic track, your goal is to do a postdoc, get your own lab, and be an independent investigator. But I wanted to make medicines, and I wanted to help patients and learn that translational science. And so my boyfriend at the time, or fiance at the time, he was still there, and he was an MD PhD student at UCLA, and he had some more training to do. So I knew I wanted to stay in LA, and I wanted to do a postdoc. And I really was fortuitous. Charles Sawyers' lab was right there at UCLA, and he was doing that translational research that I was super excited about. And, he's a physician scientist, and his lab was focused on studying prostate cancer, and really trying to understand how the mechanism of how prostate cancer develops, and how we could identify biomarkers to understand which medicines are better to give to which patients. And so I approached Charles and said, hey, I need to stay in LA at UCLA. And he knew me through the academic stuff. And he was like, oh my gosh, you want a postdoc? Yeah, come on up. And so I really kind of lucked out. I walked into that lab. And honestly, I can't say enough good things about Charles. I mean, he's an amazing mentor. And I had a wonderful experience there.
Very cool. Zooming out and just thinking about UCLA, and you mentioned going in industry was at that time probably not the popular route. It sounds like his lab was kind of a kind of more entrepreneurial lab in contrast to the rest of the campus.
Yes. Yeah. And he had a lot of physicians in the lab that were doing postdocs. So they were, obviously, physicians seeing patients, but they were stopping and coming in and working in a lab to understand how to do that translational research as well. And I really liked that experience. And it was just a sort of not as basic science as all the other labs. It had that element of application to patients.
Were there like companies like spinning out of his lab at that point in time?
Not at that point in time, but they have spun out of his lab for sure. And while I was there, we were working on a program. Oh, so when I was there in his lab, there were two things that happened. He was collaborating with pharma to develop what is now Gleevec for CML. And at the time, he was, working on the clinical trial for that. And so that was, influencing the lab, and sort of part of the lab was working on CML and the other part of the lab was working on prostate cancer. I was working on the prostate cancer, but I could see how he would bring back samples from patients and we would analyze those samples in the lab, and we could actually figure out how and why those patients were or were not responding to the drug, and helped in the development of that, of Gleevec, which was super exciting. And then I was always intrigued by that, and I wanted to kind of use that as I, advanced my career as well.
Very cool. I had like a similar experience. I was in a lab that was doing very basic research, and I was like exactly. I was just like, I want to get into that kind of translational space. And at that time too, Berkeley was like very much just like industry, like just like stay away, like stay away. And like I always like remember as I was like trying to find my like kind of translational opportunity, I was like kind of speaking to PIs, and there were some like nasty conversations that were had. I was like, I wasn't, by the way, and I was just like so naive to not like expect that this kind of like reaction would happen. And I was like, oh my God, I'm so sorry. I apologize. I am so sorry for even thinking about doing this.
Yes, exactly.
I'm so sorry. But then you fast forward now and it's more of a gradient rather than this binary thing where you're like 0 and 1. You like there's kind of like this in between. There's like the collaborations, and now companies are actually like spinning out, which was like a foreign concept back then. So very cool to see that you were at the forefront of that for UCLA.
They actually just brought me back a year and a half ago. I went out and gave a talk to my old group, and it was really fun to see some of the professors, and I was showing them all the stuff I had done at my last company, and they were actually showing the grad students. They were actually beginning to teach the grad students that there's more than just one path besides, going down academic track. There's industry, there's venture capital, there's IP and legal and business and all the pieces that come together. And I think, some of those kids were really excited to see that, it's not just so black and white as you were saying.
Absolutely. And I still remember the existential dread I had. I was like, what am I going to do? I was like you said, I got to pay some bills somehow. I got to do that. And I was like, for me, I don't got what it takes to be a professor, which is like the NBA. I'm not there. Yeah. And there's only a fine. And I was like, am I going to be destitute if I don't become a professor? This is hard. But I love hearing that they're now you go back to UCLA and they're actually trying to talk about there is the horizon is there's way more opportunity out there, because I think I try my best to stay in touch with folks who are in graduate school doing their PhDs and postdocs, and just stay close to that, and hearing that it's way better than way better, because now they're not, that existential dread is the heat has gone down a little bit. They're like, okay, there are opportunities outside.
You don't feel guilty by saying that you want to go into industry. I was really careful who I would say that to, and I can't tell that professor I'm doing that. They
I wasn't so careful. I wasn't so careful. I thought about it, and I was like, in my head, I was like, I went, I took some classes at the business school at Berkeley, And they're like, you got to do market research.
Yeah.
Like, and I was like, okay, this sounds like market research. So I'm like, let me go do this market research. And I was like, oh God, like, maybe I shouldn't have done this. I'm so sorry. Like business people, like, why'd you tell me to do this?
Yes.
But sorry, I digress. And so if you're looking, like, if you're comparing your postdoc experience and your PhD experience, it sounds like it was more basic research during your PhD.
Yep.
Postdoc became, more translational, kind of more in your pocket. Did you know after your postdoc what was next?
Yeah. So I knew, like I said, I had told Charles that I wanted to go into industry, and he was like, okay, and he was branched into industry because he had connections, obviously. But my husband was still finishing his training because he can't stop going to school.
Yeah. That sounds about right. Sounds familiar. Yeah.
So we moved to Colorado for 1 year. He was doing a fellowship after he finished his residency at UCLA, did a fellowship there. And I worked for that year as a consultant for the Mouse Models of Human Cancer Consortium. But I knew that I was moving east to the East Coast and to Boston, and I wanted to get into industry. So I was kind of, working, keeping my foot on the gas for some of the research that I'd still been doing with Charles and all the academic collaborators that we had built together. And then I was applying for jobs in industry, and I got a job halfway through that year in Colorado at Merck. So that was exciting to me. I felt like I had landed. It was it. I had my dream job, and I was in industry, and I was going to go make all these drugs and cure cancer, and off we went.
Awesome. Well, I guess the first question, how was the move from LA to Colorado? Personally, just how was that for you?
That was easier. I mean, we knew we were there for 1 year. We lived in Vail, so that's not bad.
Not bad. Not too shabby.
Yeah, not too shabby. And my little sister lives in Denver, so
amazing.
I felt like it was fun to be there. And we had 2 small children at that point, and I had my 3rd child there, and so it was a little hectic, but it was kind of a nice sort of segue into the next phase of my career.
Cool. And so you land at Merck, you're like, this is it.
Yeah.
You compare, what is, one, I guess maybe first, how did that opportunity come about? Were you just went knocking on doors? How? Yeah.
So actually, how did I get that job?
No,
I think I was connected. Someone had connected me to it was probably through the Mouse Models of Cancer Consortium. Someone had connected me to the hiring manager at Merck, and I applied through that. So it wasn't like a cold into Merck, but they were doing the translational research, and they were looking at mouse models for understanding biomarkers and how you could use them to track efficacy in patients, which was right up my alley. It's what I had done in my postdoc. So it was a really good fit. And I learned a ton there about drug discovery, but it's definitely different than what I had been exposed to.
Totally. And before you got to Colorado, I know you were a professor for a little bit. Was that after the Colorado experience or before?
Yeah, no. So it's sort of a weird in between. So I did my postdoc. I was a postdoc for 3 years with in Charles's lab. And then I was an adjunct professor for almost 3 years while my husband was finishing his training. And then I went to Boston, or to Colorado, and then to Merck.
Got it. Very cool. And we were again, kind of before we hit record here, talking about, some how networking and this meeting people is important when it comes to kind of securing opportunities, which was for me a very novel concept back then. And I can't pretend that I'm good at it now, but it's kind of something for anyone out there who's trying to figure out what that next step is critically important that you put yourself out there.
Yes.
Even though it is a little bit scary.
Yeah. And Charles was really, I think I learned that the most from him. He is probably one of the most collaborative scientists I've ever worked with. And, he does top-notch, state-of-the-art research, top-tier journals. But rather than, people can get, oh, I'm going to have the Nature paper and, I'm not telling anybody what I'm working on.
Yeah.
His approach and philosophy was very much like, well, if they're doing something similar, let's collaborate and do something better. Right. And that's like his way. And that's how I was trained. And I really like that. I think I flourish in that sort of environment. And that's how I began to network with folks. And so I made a MIC model for prostate cancer there. And some other labs had an AKT model, some others had a PTEN model. And instead of us all racing to fight each other, who figures out prostate cancer, we were figuring out and comparing our data so we could understand what makes this mouse different than that mouse, than this mouse, so that we could actually advance the science. And through those collaborations and networks, I really, gained access to a lot of really exciting research and people that really helped me along my career.
That's incredible advice and like perspective, because I think it's kind of like this thing where if you just feel like, if you think of it as like a thought experiment, if you don't like reach out, you could just be doing redundant work.
Yeah. It's a waste of time.
Yeah, it's a waste of time. It's like, yeah, I get it being first author, publishing in Nature and being first, there's like something to that too. Yep. But you're just like, thinking about it, if you guys just never speak, you could be just like, be like in these parallel tracks and you're just like, come on, we would be far more like productive together, which I love to hear, because, sometimes it's kind of like you have to like swallow your ego. You're just like, yeah, we could do this better together. So it's very cool to hear. And so, thus far, before Merck, you've had a lot of like academic laboratory, and this sounds like obviously the first big industry opportunity and one of the biggest. Can you talk a little bit about like, what that experience was like first day, like early days in a massive organization like Merck? What was that like? Did you have like culture shock, or was it like, oh, this is it?
Yeah, no, it was definitely a culture shock. Just because it is, I think, those big companies, and you have to be, because they're so big, they end up being very layered and bureaucratic and sort of siloed in functions. And that was kind of the exact opposite of how I'm used to working and where I thrive. So that to me took a lot to get used to. And I didn't really understand that it was a little bit more like kind of mass production science. Like you do something and you hand it off to somebody else and they take it from there and then they take it from there and it keeps going, which I thought that just was foreign to me. But that being said, I think what I did learn, and what I think industry obviously is exceptional at doing, and big pharma, is really drug discovery, basic drug discovery. And, I think I've said this all the time through all my career in industry, there's two tracks. There's the basic science, where you need to really understand deeply the biology as to what's driving the disease. And then there's the drug discovery aspect that's layered over that, where you take that knowledge that you've understood about the biology and you apply that and you go through the process of developing a drug. And I learned that process of developing a drug when I was at Merck. And so that was extremely valuable to me. I feel like I came in with kind of the understanding on how to do the science part of it, but I learned and layered on top of it the ability to, or the process of drug discovery.
Very cool. And with I'm going to imagine the drug discovery process at Merck is one very rigorous and a very large endeavor.
Yes. Yes.
And despite the silos, did you, when you were learning, were you able to kind of pick the brain and tap on shoulders of folks who are like, let me show you,
Yeah, for sure. And I think, and then again, I was thrown into the deep end in that setting, right? Because I didn't know drug discovery. I didn't really understand the process of it at all. I was in a lab and doing basic science. Right. I sat as a core team member on one of the main projects there, and I was the biology lead. And then there was DMPK and chemists and biochemists and, all kinds of functional heads that came to those meetings. And I got to see all of those components that go into developing a successful drug and learn from each one of those people along the way, which I think really, ultimately that type of big pharma environment wasn't the best fit for me, but that learning experience at Merck on how to do that part of it was very valuable as I took my next step into Agios.
Yeah. And like, I think about that. It's kind of, it might not be your forever home, but like, it's like, what an opportunity to have, like, to be at the table with like all these experts. Like, where else are you going to find that, right? And then you can just like osmosis, just like soak it up. And I always think about whenever I'm trying to learn something new, I can like, it's almost advantageous to like come in with a blank slate, because people are like, I got it. Like, you have like this window of like 90 days to like just be like, to be annoying, basically.
Yeah.
And where they're not annoyed with you. You know, like, you can, there's like, of course, you're new. Let me show you.
Yes.
But like, if you go past like 90 days or whatever that date is, they're like, come on, you should know this by now.
Figure this out.
Yeah. You got to. So I always like, whenever I'm like new to something, all right, I was like, I have this window. I can ask all the annoying questions now, and then just like start absorbing.
Yeah. For sure.
Yeah. So, you know, it sounds like you were able to really just like learn a lot about the drug discovery process. How did you land? You mentioned like you decided to move to a smaller company. How did that opportunity come about? And talk a little bit about that experience.
Yeah, I have a funny story about that. I remember Lou Cantley, who was one of the founders of Agios. His lab was at Harvard, and it was right outside the door of Merck. The buildings sat next to each other, and he came over to Merck one day and gave a talk about basic science, but how these genes, these metabolic genes, could be playing a role in cancer. And I just thought that was so interesting to think. Everybody at that time was looking at all these kinase cascades and how they are going to drive cancer. This was just like a new way to think about it. And so I was really intrigued by that. And I actually got a cold call in my office. I'm not even sure how Michael Sue got my number, but he did. And he said, hey, I heard about you. You have a lot of experience in, molecular biology and animal models and translational research. We have this new company, Agios, that we're starting. We'd love to, interview you. I'm like, okay, I didn't know anything about it. And Lou had just spoken, so I was excited. So I went over and I interviewed for the job. There was not even, they didn't even have labs yet. Like, we're sitting on stools. Like, it was like this gutted place. And I'm leaving the gorgeous Crystal Palace of Merck to go over to like this like warehouse room.
Yeah. I love
and
that.
I was like, what's going on here?
Yeah. Am I going to get kidnapped? Like, am I safe here right now?
Yeah, and I had no idea about the whole biotech thing. Like, I was very naive to that. And so I came back and I called Charles Sawyers, and I said, look, I've got this opportunity. I don't know anything about it. What should I do? And he said, I don't know. Let me call Lou Cantley and I'll call you back. And then he called me later and he said, well, I think you should join the company and I'm going to join the SAB. And I'm like, okay.
Wow. Wow. Now, what a like kind of great way to back channel, and such a good cosign. Like, not only do I like what I hear, but I'm getting involved.
Yes. So I was like, all right, enough said, I'm in. And then I left.
Very cool. And what timing too, that like there's a talk and bring like just a cold call. And I love that approach too. Like, it's like I was like talking about like just like how do these opportunities come about? Sometimes it's just that like it's that cold call, or it is sometimes it's like mustering up the courage to like just reach out. And I understand like that is a hard thing to do, but sometimes that's just like it sounds like this was a pretty big pivotal moment, from a cold call.
And I was scared, right? I mean, you have a job security, and I, had 3 small kids at home. My husband's a resident. He had just finished his fellowship still, and he was just like running around, like we're just getting started. And, I was like, oh my gosh, am I going to really like just go to this tiny little company that, at that time they had raised $30 million. That was $1 gazillion back then. I mean, that was nothing, but back then it was a big deal. And, I was worried that maybe I was making a mistake, but I think to your point, I got thrown again into that deep end, but I was in that deep end with everybody in that company, and the collaborative spirit and the everybody roll up your sleeves, we're going to work together, was the piece that I was craving that I didn't have as much at Merck, and so I felt like it was like the perfect opportunity. And now I learned the drug discovery. I have a good handle on biology. I can figure out how to do experiments. And then the other part of being in a small biotech was that I got to see the business side of it up front and center. And that was really exciting to me. It was like learning all of the pieces that go into building a company. I've been learning all along in my career, and I didn't realize it.
Yeah.
But it was happening.
Yep, absolutely. And when you joined, how big was the team?
I was the 11th employee at the company.
Oh, wow. Okay.
Yeah,
Like
I know.
very nascent stages, very nascent stages. That's so cool. And so I know your first role was, director of pharmacology. Can you talk a little bit about what were your responsibilities? Obviously, I'm with a startup. I'm going to imagine you wore a lot of hats, probably outside of pharmacology as well. But can you just talk a little about that era?
Yeah, so they brought me in because I had a lot of experience in mouse models, and we were trying to use mouse models to, as a more sophisticated way to study cancer and the diseases associated with cancer. And, so I was in charge of all the in vivo kinds of studies that were going to go on. So we had a biology team that did the in vitro work. And then when it moves into the next level, I was the in vivo biologist that was going to take it in that translational science, again, bringing together like the DMPK and the drug properties and all these things so that we can, biomarkers that you need to drive drug discovery. So that was really my initial role. And then I was given the opportunity to work and lead the IDH program, which was the lead target lead program at the company. And I headed up the translational biology team, and that was kind of pulling all of those functions together and then driving the science for the discovery of those molecules.
That's so cool. Also, like, what an opportunity. Just like, they're like, here you go. Like,
yeah, it was really amazing opportunity. And again, I had the team around me was just, supportive. And, I think I learned at that stage, it's always scary to say you don't know something, right? And, part of throughout your career, you never want to admit that because you don't want to look dumb. But, when you're some point, you get thrown in so deep that you're like, all right, look,
yeah,
I don't know what I'm doing. I need but I can figure this out if we work together. And that was kind of the mentality that Agios had. And everybody just rolled up their sleeves and we worked together to push those molecules into the clinic.
I can go in so many directions, but something that really stood out to me as you're describing this is like, again, it's kind of like, it's like kind of scary to admit that you don't know something, but
Really
it's so powerful. It's so powerful.
It's freeing for yourself too, right? And I think you make that leap when you are willing to admit that. You make a leap much faster, and you put your ego aside and figure out how you can synergize with other people to drive things forward.
Absolutely, and I think too another aspect of that is it's like the secret that there is no secret. We're all just figuring it out. Like. We're all just figuring it out. Like, and sometimes you can kind of see like Agios is like a very large like company. It's kind of, or you look at like NVIDIA now and you're just like, oh, they have it figured out. It's like, oh no, they were in the salt mines for like 30 years figuring it out. And it's like everyone is just figuring it out. And like part of that is just, and that, like you said, it's like liberating. You're just like, okay. So we've put the egos aside. Can we just like, let's work together here?
Exactly. And that for me was like the biggest, and that came from the top at the leadership at Agios. They really empowered the teams to think that way and work that way. And, our CEO would walk in the room, our CSO would walk in the room. We'd have RAs presenting. No one batted an eye. No one cared. It was more of like, just get up and talk about what your research is and what problems you're having, what things going well, what's not going well. And we can all collectively kind of think through this problem to help advance things. And again, I think that was a unique opportunity there with that environment and that culture that was set up. And that came a lot from David Schenkein. That was his MO. And I think really is what drove the success of the company.
And I love to hear that too, because like sometimes people talk about like company culture is kind of like this soft, like kind of nebulous, fluffy thing. And we're scientists. It's like, we're hardcore, like we're super objective and like quantified, right?
Yeah.
But then like you think about this, like all of this is possible because of that soft, fluffy, nebulous thing of culture and like being like intentional in its design.
Yeah.
So I love hearing that, because like also it makes people want to stay and work at your company.
Absolutely.
Like that's important too.
Yep.
And so, in this role, I know you also were managing collaborations, like external collaborations with academia and CRO. So maybe if we can unpack that a little bit, how, you're like, okay, you're now in industry, and now you're like, oh, like my colleagues in academia, like how do we design an effective collaboration? How did you approach that?
Well, that was to me like super natural, because I had a lot of big network in academia because of, I'd spent so much time there. And like for me, when you do making a mouse model, a lot of these mouse models take years to generate. And so like in industry, you don't have time or the money to like go and remake the mouse model and then do the experiment. So I went to my boss and I said, look, Kwok Wong has the right lung model I need to go study. I'm like, what do you want me to do? Do you want to spend 2 years making a mouse model?
Yeah.
Or I'm gonna just give him the drug and let him do the experiment and we can move forward? And luckily, again, the team and the way the company was set up, everybody was like, well, no, we'll just figure out the way to, give them access to the molecule. And it's a win-win, because companies know how to make drugs and potent drugs that they understand how to dose and use. And then the academic labs, that's what they're lacking. They have the deep mechanistic understanding, but they don't have the pharmacology tools to be able to dissect the biology. And so that synergistic relationship between those industry and academics is a win-win. And we always co-publish together. They publish, we advance the programs, we get to the clinic, and they get to, write grants and be part of these really exciting academic industry collaborations that bring drugs to patients.
That's so cool. And I always in my head, I was like fist pumping in my head. I was like, that's exactly how I think about like partnerships, is like these like positive sum win-win situations. And also it just reminds me again, just kind of this thing where the collaborative nature is like super important here, where you're like, do we recreate the mouse model?
Yeah.
And just like these, like do the same work again, recreate the wheel, or let's just like keep it moving. Like let's keep it moving.
Absolutely. And we had a really great lawyer at Agios who was like, of course we're going to make this work, right? I mean, they're not going to get hung up in the legal jargon. I mean, it takes a while to get through that, but we were willing to move quickly and figure out ways to be creative with the IP around what's generated, so that it's, companies protected, the industry or the academic labs are protected. And we collaborated with multiple groups. And I always do that. And then the way I collaborate with them too is I'm very transparent. Like I'd say, oh, I'm working with you on this part of the project, and I'm working with X lab on this part of the project. And then we would have lab meetings with all three, the industry and the 2 and 3 labs together, so that there's no surprises, no one's mad. Everybody understands where the authorship's coming right from the get-go. And then we parse it out into multiple papers. And I felt like, that really worked well for us there. And I use that today in the company that I've started.
That's awesome. And I think too, as like I'm hearing you kind of, there's like a lot of intentionality to it too. And it's also like empathy, where you're like, I need to understand what you are motivated by and how you are feeling. And if you just ignore that and just like bulldoze them, that's like, not a great, it's more zero sum. It's more zero sum than it is positive sum. And I always like try to like talk to my team and just like put yourself in their shoes. Like what is it that they care about, and do your best to ensure that they get what they care about. Yeah. Obviously not at the necessary expense of you, but it's always like is an exercise in empathy, which I love hearing. Now, okay, and that's academic collaborations. Now, collaborations, not academic laboratories.
Yep.
How did you approach that?
Yeah. So again, at Agios, we didn't have a ton of money. We had some lab space and we had teams that were working and doing work. But actually, we had a woman there, Shen Fang, who had a lot of connections back to Chinese CROs, which at the time were just starting to get up and running. And the labor, obviously, was a lot cheaper over there. And they were trying to build CROs, but they didn't have the technical expertise, whereas the people in the US had the technical expertise. And so it was a win-win where we built a lot of these collaborations with some of these initial CRO groups to, provide guidance to their team to help them or help us move our projects along. They also, we treated those team members, though, as part of our team. We didn't throw something at them, be demanding, oh, I want to see this assay. We explained the science, why we were doing the experiments. They would come to our meetings. So they felt like they were part of the team, and that it was more rewarding for those employees at the CROs to be part of our projects. And so we would get the top tier people on our projects because they were excited to work with us because we didn't treat them like slave labor, right? And so I think, again, it just goes back to that theme of being collaborative at every stage of the place. And, we had CRO work in the US too, different places for different activities. But again, it was always treating people with respect and treating people as a group.
Totally. And I love hearing that because for Exceeder, I've been on the other end where it's I just been squeezed really, really hard. And I get it. That person doing the squeezing, you got a lot out of me. Yeah. But it's a terrible feeling and it does not make me want to work with them again. And life is long and you got to think about, at least for me, look, this is I'm speaking for John here. I'm not going to speak because there are very large institutions that get by squeezing everybody. Yeah. And that's just that's their culture, but it's not mine. Because it was a very formative experience. I was oh man, this is this is awful. And then from then on, I was exactly what you said. Even though they're a vendor partner, I treat them part of the family. This is
exactly.
Exactly. Just a very specific tactical example is when people send us bills, we'll pay it early. We'll pay it early because we know that money in your pocket helps you. And,
exactly.
And versus, there are large companies who are I will pay you in 120 days. And maybe I won't. Maybe I'll miss that payment. Yeah. And then, for your partner's I kind of needed that money to pay my staff or whatever. Yes. And that experience can be really, very tenuous and painful. So I love hearing that, despite being a vendor partner, it's hey, come on in. We're going to be collaborative. You're going to learn too. So it feels like a much more healthy relationship.
Yeah, for sure. And we definitely, we worked hard. We went on trips. We would have personal interactions with the team. We would go over there. They would come over to visit us. They didn't have Zoom back then, but we would have late night phone calls, and sometimes we would be the ones staying up late and not making them stay up late, right? And so we would just rotate, and I think it worked for us. And Shenfang did an amazing job of really managing all of those international relationships.
That's amazing. And I realize this is the baptism of fire era for you. I know you also opened up and led a vivarium as well.
Yeah. Yeah. I changed rat cages or mouse cages, I should say.
Can you talk about that experience a little bit?
That was crazy. No, we did open the vivarium in the site. I brought my technician. I stole my technician from Merck, who was wonderful. And she had a lot of mouse experience. Literally days I was washing cages, and we would go in on the weekends and feed them and change the bedding, and we didn't even have the staff to do that. So it was we were soup to nuts doing the whole thing.
Yeah, that's amazing here with you. You were talking about the layers of kind of skill sets that are being acquired, basic science. translational drug discovery. You're doing like these soup to nuts, the in vivo. Like nowadays, you're like you're giving Charles River a call, like,
Yes.
hey, can you run the studies?
Yes.
But no. Like you were like, we're gonna do it. Like
Yes.
we're gonna learn how to do it. And I think there's something about that I think about for myself too, is that you learn a lot by just like rolling up your sleeves and getting your hand dirty. And I think something about that too is that once you do that, you're much better at leading and managing a project that involves that, because you're intimately aware of like
what it takes,
what it actually takes. And that is, I think, it's always like, I always say like, no one's above anything like at this company. Like there's value in just like rolling up your sleeves and getting your hands dirty. And so I love hearing that. And, and now again, it's kind of like, with it, I think Charles River already incorporated Explorer Biolabs, but like Vivarium, like you can just like, you can almost do like a WeWork for your rat studies.
Yes. Yeah. I don't think many people have live vivariums anymore.
Not anymore. Yeah. Not anymore.
But, and it's really expensive, and there's a lot of paperwork and stuff that goes along with that too. But at the time, it was what we needed, and we rolled up our sleeves and we got it done.
Thanks for joining us on this episode of the Biotech Startups podcast with Kate Yen. Be sure to tune in for part 2, where we explore how she led groundbreaking cancer research at Agios, tackled the challenges of advancing a novel therapy through clinical trials, and ultimately built Auron Therapeutics from the ground up. Kate also shares insights on navigating biotech fundraising, structuring partnerships with big pharma, and the importance of staying lean while building a successful biotech company. If you enjoyed this episode, subscribe, leave a review, and share it with your friends. See you next time. The Biotech Startups podcast is produced by Exceeder. Don't want to miss an episode? Search for the Biotech Startups podcast wherever you get your podcasts and click subscribe. Exceeder provides research labs with equipment leases on founder-friendly terms to support paths to exceptional outcomes. To learn more, visit our website www.exceeder.com. On behalf of the team here at Exceeder, thanks for listening. The Biotech Startups podcast provides general insights into the life science sector through the experiences of its guests. The use of information on this podcast or materials linked from the podcast is at the user's own risk. The views expressed by the participants are their own and are not the views of Exceeder or sponsors. No reference to any product, service, or company in the podcast is an endorsement by Exceeder or its guests.